Thank you. Good morning. So it's great to be here. Thank you, the organizers, for giving me the opportunity to show off our work. So in my group, we use theoretical and computational tools to understand collective behavior in biological tissues. And I'd like to show you some of our ongoing recent work from this area. So starting with simple epithelial tissues, which is two-dimensional covering that is basically everywhere inside the body. Whenever you have lining and surfaces, they form that protective layer which protects you from the outside harsh environment, not letting anything through. Under normal conditions, think about just say your skin, they really also protect you from mechanical deformation. They don't easily flow just because you tug on it or you pull on it. They are static. The cells are linking with adherent junctions, and they form these tight structures that really can resist deformation. I would say under, say, some abnormal conditions, you can, I'm sure, think of many, but one artificial one is just you artificially, say, cut your skin, created a wound. There is, of course, a collective flow of cells in response to heal that wound. And from a physics, sort of mechanical materials perspective, you can think of this almost as a transition that goes from a static quiescent state, which it has, under wound healing, has been triggered to flow collectively much more like a liquid or fluid type of state. It is then therefore very important to understand, how do tissues behave in response to external perturbation, and how do we approach it from a sort of a physics and mechanical perspective. So I just want to show one recent, really simple yet interesting experiment by just trying to answer that idea by just pulling on a piece of tissue, right? So this is cells that's grown on a substrate, MDCK cells that have been lifted from the substrate that they were grown on, and simply just trying to stretch it to measure the mechanical response. This is work from the group of Guillaume Charas at UCL, where they just essentially measure the stress versus strain response. And one thing that you can already see is that there's quite a bit of nonlinearity in the response, right? Where the stress does not go as a linear function of strain, it actually bends. So you cannot simply describe this with as a Hookean solid. And just to give you an extreme sort of example, if you thought the last one was a bit artificial, there's recent work from the group of Prakash at Stanford, where they looked at this simple animal. It's really an animal it's called trichoplax adherens. It's made of only 2 epithelial layers. And these cells are motile due to having cilia on the bottom. And over time, over actually almost real time in the movie, they actually exert so much motility force inside the layer, and so much so that you can actually have like breaking or fracturing of the whole tissue due to these active forces. And you can almost think of this also as a mode of asexual reproduction. But what's interesting to me is from a mechanical perspective, this is highly unusual mechanical response in that it's almost like a brittle material under certain situations, or ductile type of fracturing. So if you go look at this very interesting paper, it has a whole collection of behaviors like opening up a hole, but also be able to heal that wound over a short amount of time. So as a material, it's a fascinating system and just highlights how tissue mechanics can be really rich. So I want to start by our very simple sort of physicsy model, trying to understand these systems. And you may have seen this over the years. This has been a class of models that's really simple, trying to describe cells as essentially 2D cells as polygons inside a layer, except you prescribe certain biomechanical interactions that govern how cells interact with each other. So this has been around for a couple of decades. And the idea is you write down a work function or sort of an energy functional that governs how cells interact with each other. So here A is the area of each polygon of each cell, and there is elasticity governing the fluctuation of the cell area, and then a second term captures the cell-cell junctional dynamics. So there's an adhesive energy as well as a contractile tension due to contractility of the cell. And then finally, there's elasticity coming from the actin-myosin cortex, which is essentially, think of that as a rubber band which is wrapped around the perimeter of the cell. So PI is the perimeter of the cell. And I can turn this even into a much more simpler form by non-dimensionalizing, assuming all the cells behave similarly. And you have this, finally, sort of a very geometrical interpretation of how cells behave. And essentially, it's going to be a polygon which prefers to have a certain area. At the same time, it prefers to have a certain perimeter. So these will be the parameters of the model that we can study. So one thing that we have done recently is to simply just shear a tissue, a model tissue like this. And here is a movie of what is happening. So I'm applying simple shear at the boundary, and the stress is evolving as a function of strain, pretty similar to that experiment that I showed earlier. And what's different also is that in this model, because the cells are confluent, right, they're not opening any holes, there's no gaps created, they really have to accommodate a shear when it's large enough. And they do this by these sort of large-scale rearrangements, so-called T1 transitions, but they all happen at once, and you get this large stress drops that's akin to sort of avalanche type of behavior in the strain trace. Just to connect a bit to experiments that have already taken place, actually there's a very nice experiment from the group of Beth Pruitt at UC Santa Barbara, where they actually do this kind of simple shear on a tissue, on an MDCK cell monolayer. So what do we find in the model? Are there any trends when you shear it, right? So one thing that I'll show you is the response of the tissue to shear as a function of this preferred perimeter P0. Remember, that's the geometric interpretation. And in fact, what we see is that if you tune this parameter P0, which is dimensionless, you can non-dimensionalize it by dividing by the square root of the preferred area, there's actually a nice phase transition that takes you from a solid-like response to a fluid-like response. And let me show you what that looks like. In the solid state, this is almost any solid, right, would do that. If you shear it, it will just respond by going up in stress, and you can capture a mechanical shear modulus, some kind of response, right, telling you that it will cost energy to do that. And what's interesting is that in this model, we already see a lot of the nonlinear elasticity that you saw in those experiments I started with. And also, what's extra are these intermittency due to the plastic rearrangements, these sort of avalanche dynamics that I won't go into today, but if you're interested, I'm happy to talk a lot about it offline. So what's also quite interesting is what's happening on the liquid side or the fluid side of this model. By definition, if you shear a fluid, it does not respond, right? That's the definition of fluids. They don't have a shear modulus. But interestingly, if you go far enough, if you strain far enough, if you shear far enough, the stress actually starts to build up. Not so different from what you see on the top. It has also this kind of avalanche dynamics. So we see that it goes from a fluid to a solid purely as a function of strain, okay? And there's sort of a critical strain where this happens. And people studying, say, fiber networks, cross-linked fiber networks, have also seen something like this, and it's so-called strain stiffening. But essentially is a stiffening, it's a rigidification of the tissue that is purely driven by the strain itself. And we can do this systematically at all these different values of this preferred perimeter. And you can sort of map out a phase diagram which tells you how much strain do you need in order to rigidify. And we do quite a bit of work in trying to characterize as physicists what's the nature of that transition. Is it sort of a second order phase transition? Are there any analogs that you can see in even non-living systems? And yes, there are. We can do sort of a critical scaling to show that it's a second order phase transition. But one thing, I want to just take the next few minutes to tell you about sort of a very simple mean field yet satisfying explanation for why there is these type of transitions inside tissues in the first place. So this is a mean field description of a single cell, of a single polygon in the model. And I just want to, maybe let the whole audience think, right? So if you have a polygon with, say, a regular polygon, that would have area 1, and so a square would have perimeter 4. And for this shape, are there any ways that I can deform? Can I deform this cell without changing both the perimeter and area? So raise your hand if you think there are ways to do that, to accomplish deforming this without changing P and A. Okay, there's a few hands there, right? But so I should caveat, the caveat is this doesn't include the trivial rotation, which is an obvious way. But can you kind of distort it into like a diamond-shaped parallelogram? So if you actually try to think of ways, there's actually no ways to do that. And one simple reason is that this is already the minimal perimeter shape for the given area. So anything you do to it will change those two quantities. On the other hand, what about a slightly elongated cell, something that's non-regular? So are there ways to do it? And you may see the answer coming. There indeed are many ways of deforming this without changing P and A. So why do I have this exercise? Well, if you think about the energy that I introduced, the shape of the cells are governed by the perimeter and area. And if I find a way to deform it without changing P and A, I'm essentially not changing the total energy, and therefore I'm not putting on any forces on the cell, right? So that should come freely if I can find ways to do it. But if I can't, that means P and A will change if I deform, and the energy must go up, therefore the stress must build up. So that's sort of the connection. And you can formalize this a little bit, really trying to visualize what are the ways to deform a single cell without changing its energy. And here are just the animation showing you what are the family of shapes that do not cause any energy. And so this would be the fluid-like state that's in the model, which says that, well, I can really deform up to a certain limit without having the stress build up, which is the definition of a liquid. So I can, go back to the energy function. I can rewrite it, but essentially here's a pictorial description of what's happening. On the fluid state, you have these ways to walk along the energy landscape without going uphill, and that's liquid-like response. On the solid side, anything you do will force you to go uphill, which therefore gives you a solid-like response. Okay, so I see I have like a couple of minutes. So one thing that is ongoing, you see that so far the work has been two-dimensional, and we are now really extending this to 3 dimensions and trying to think about really physiological relevant, not just a simple monolayer, but multiple layers that, for example, you see in the epidermis. And this is supported by the Human Frontier with my collaborators, Tamal and Friedhelm. What we're doing is we're building these 3-dimensional vertex models to think about what happens if you have stratification, what happens, leads to stratification, for example, and what are the mechanical forces that are involved in this whole process. So Friedhelm especially is the expert on measuring the rheology of forces and forces inside the tissue using these microdroplets. And then finally, I'll just show one ongoing result. This is from my postdoc just a couple weeks ago, we're not just studying stratification, which is going from one layer and built into multiple layers. In the experiment of the organoids, we actually see the process of the reverse happening. So something that's already stratified actually can condense down into a single layer. And I just want to show you what happens, what we think could be happening from a simulation of a multi-layered vertex model. So this is under mechanical compression, but also internal activity. And you see that it's essentially being forced into a single layer, intercalates, but also undergoes something that looks almost like fracturing. Okay, so I'll close by thanking my students, postdocs who did all the work, and also the Human Frontier, especially supporting us on this new adventure in measuring the rheology of epidermal structures.
Thank you very much, Dapeng, for the very interesting presentation. Are there questions? We have time for a couple of questions. This one there.
Hi, it was a nice talk, thank you very much. I was curious, presumably signals of something that's happened to the tissue, like a wound has occurred, are being sensed by the cells at the periphery, but are the other cells responding in the same way? I mean, or is it just the peripheral cells that are making these decisions?
Yeah, so in the wound healing scenario, cells at the wound edge are quite different from what's in the back. So they are sort of the pullers, and they also secrete signals that tell in the back to push, actually. So there's pullers and pushers. And also, I think that also leads to, say, compensatory cell division that happens towards sort of deeper inside the wound.
Cool, thank you.
There was another question there.
So I have two questions. So one, in the solid-like tissue, you showed that there is a avalanche dynamics that happens. So is there a self-organized criticality associated in the system? That's one question. Second is, in the fluid-like state, you showed that there is a symmetry associated that does not change the energy. And it's reminiscent of the Goldstone boson.
Yes.
Are there any Namo-Goldstone modes in the system? And what are the broken symmetries in the problem?
So I'll maybe answer the second question. So these indeed are akin to Goldstone modes, and these are essentially just the zero modes in the energy, and there's a few of them with respect to certain deformations you can have. So in this one, if I just have the so-called affine deformation, right, there's really basically one mode with which you can move along without changing it. So that's exactly the Goldstone mode. With respect to the avalanche, yes, indeed, it looks like it shares a lot of similarities to avalanches in other systems, and we are still sort of trying to, get a good exponent to describe the avalanche dynamics, but it does look like it's similar to, say, the sort of global exponents that people see in, say, avalanche of granular materials. And essentially the idea is, right, one cell undergoes a local rearrangement, and that dumps a bunch of stress on the surrounding cells, and those cells can be made to go over the threshold for them to rearrange, and it sort of cascades in the system. And there's some also experimental evidence we are pursuing in systems that are very motile, and they have motility-driven rearrangements.
Okay, so thank you very much.
Thank you.
We proceed.